I'm Dr. Kimberly Steele, and for those of you who don't know me, I'm a parent of two boys, Michael and Matthew. Michael's the oldest and was diagnosed with KLA. And I'm also a surgeon scientist and have come to the LGDA organization as a parent and also as a physician and scientist to help. And so I'm really excited and appreciative to host this series. This is the LGDA Presents the 101 series, and it's the basics of complex lymphatic anomalies. And why are we doing this? We're doing this because we've got an international conference coming up. It's a scientific conference, and it's October 2nd and 3rd. And we would like to have our community prepared for that conference. So we have already done 2 of the webinars. We've talked about genetics, and we've talked about the diagnosis. And we will, on August 30th, talk about complex lymphatic anomalies as just a general topic. But today, I'm super excited to have 2 incredible scientists talk about research. So this just tells us about the goals of our LGDA series and the conference. Don't forget that date. I hope you'll all join us. It is a free virtual online session for 2 days. And for those of you who have attended the pre-conference series, you've seen this slide, but for those who have not, just to go over again what complex lymphatic anomalies are and what the LGDA is representing. It's a group of 4 subtypes that overlap in their patterns of clinical symptoms, anatomic location, imaging features, and the blood alterations. That's hematologic alterations and complications. And over here, you see the overlapping groups that are included: generalized lymphatic anomaly, Kaposiform lymphangiomatosis, Gorham-Stout's disease, and central conducting anomalies. So today, exciting, we are going to talk about preclinical models and learn about what research models are and how they're used in complex lymphatic anomalies. And I am excited to introduce, super excited, 2 important, as I said, scientists who have been the everything to the LGDA and research. There's Drs. Michael Dellinger, who's an associate professor of surgery and molecular biology in the Department of Surgery and Molecular Biology at UT Southwestern Medical Center, and Dr. Timothy LaCroix, who is an associate professor of pediatrics, associate dean of graduate education at the University of Cincinnati and Cincinnati Children's Hospital. Before I turn it over to them, I just wanted to share this personal moment. That is my Michael, and that is Dr. LaCroix and Dr. Dellinger. About 2 years ago, as we were in the midst of the diagnosis and treatment for Michael, we had the opportunity to meet these incredible scientists at a conference in Austin, Texas. And this is a picture of us meeting them. And I will tell you how important it was to meet them and how they have been such an important piece of helping Michael. And I'm excited for them, as I've said, keep saying excited, for them to share all of the work with you and you see what they have done. So if you guys want to take it over, I'm happy to give it over to you.
Thank you, Kim, for the kind introduction. Can you see my slides?
We can, Mike.
Again, Kim, thank you for the kind introduction. And I'd like to thank Kim, Lisa, and the rest of the LGDA team for giving Tim and I the opportunity to present some of our work on preclinical models of CLAs. Before I start, just with respect to disclosures, I'm the Director of Research for the Lymphatic Malformation Institute, and the LMI is a nonprofit organization that funds research on complex lymphatic anomalies, and they have funded some of my research that I'll present today. Before I get started on preclinical models of complex lymphatic anomalies, I thought I would just briefly give an overview of biological organization. And in these cartoons, they depict an organism. And organisms are made up of different organ systems and organs. Organs are made up of tissues, and tissues are made up of cells. And when we look closely at cells, you can see within the nucleus of the cell is DNA. And DNA is the genetic material. It's the molecule that contains all the instructions for the development and function of an organism. And it's made up of 4 building blocks, G, A, T, and C. And these building blocks form strands, which form a double helix. And then DNA is packaged into thread-like structures called chromosomes. And we, as humans, have 46 chromosomes in every single one of our cells. And DNA is important because it codes for proteins. And proteins are the molecules in cells that carry out different functions. Proteins can form the cell's skeleton. They can tell a cell to grow and divide. They can inhibit the growth of cells. And they can cause cells to migrate and move around in its area. So DNA provides this information for proteins. And changes to DNA ultimately result to changes in protein. So this mutation or a single change in one of the base pairs of the DNA can result in a change in the sequence of the protein and cause that protein to have an abnormal function. That then abnormally functioned protein causes cells to act abnormal and can ultimately result in human disease. And there are different types of genetic mutations that can cause human disease. There are germline mutations and somatic mutations. And germline genetic mutations are mutations that are present in either the sperm or the egg. And in this case, it's present in the sperm. So after the sperm fertilizes the egg, an embryo is created, and every single one of the cells in this embryo has that mutation. This embryo then develops into an adult human who can then pass on this genetic mutation through his germline. A somatic mutation is a mutation that's not present in the sperm or the egg. But this is a mutation that can randomly occur during embryonic development or postnatal life. And this results in a patch of cells carrying the mutation. And you can see this mutation is not present in the germline and is not passed down through generations. So germline mutations can result in inherited disorders, whereas somatic mutations can cause sporadically occurring disorders. And complex lymphatic anomalies are typically caused by somatic genetic mutations. So for complex lymphatic anomalies, a mutation can occur in a lymphatic endothelial cell. And lymphatic endothelial cells are the cells that make up lymphatic vessels. So in this case, we can imagine that this blue circle is a cell. It then gives rise to 2 daughter cells, and this cell acquires a mutation, which is then passed on to cells through cell division. Ultimately, we then have a mixture of mutant and normal cells, and these mutant lymphatic endothelial cells can form irregular or malformed vessels, whereas the normal cells can form regular appearing vessels and regular functioning vessels. So these malformed lymphatic vessels can have altered function and ultimately result in disease. So normal lymphatic vessels are the body's drains. So in our body, our blood vessels lose fluid, sort of like this hose here losing water, and lymphatics are the drains which collect that fluid and then drain it away. So under normal settings, like I said, our blood vessels are constantly losing some fluid, and the lymphatic vessels are there to pick up that fluid and drain it away. And when we have mutations that affect lymphatic development, they ultimately affect lymphatic function, and these drains no longer work properly, and that can cause fluid to accumulate in different regions of the body and cause various complications in patients. So, like I said, changes to DNA ultimately result to changes in a protein, and its abnormally functioned protein causes the cell to misbehave and form an abnormal malfunctioning vessel. And that can ultimately impair lymphatic function and cause fluid to accumulate in areas. And we're interested in understanding or identifying disease-causing mutations because these mutations can ultimately help us develop preclinical models and guide the search for therapies. So if we understand, or if we know what mutation is causing a particular complex lymphatic anomaly, we can use that information to engineer cell models or animal models of the disease, and we can use those models to better understand the molecular mechanisms driving the pathogenesis of those diseases and to test various therapies. So there are several different types of preclinical models, but the ones that we're going to talk about today are tissue culture models, a zebrafish model, and mouse models. Tim is going to handle the tissue culture models, and I'm briefly going to talk about zebrafish. And my lab primarily uses mouse models, so we'll spend more time on mouse models. So zebrafish do have lymphatic vessels, and they are frequently used to study lymphatic development in diseases of the lymphatic system. And there are several advantages to using zebrafish to model lymphatic disease. You can see here in this picture, this is an image of a zebrafish whose lymphatics are fluorescent. And you can nicely see the entire lymphatic network in this zebrafish embryo. So zebrafish are advantageous for research because the embryos are optically transparent. We can see through them, and that allows investigators to take these really nice images of the lymphatic network. They're also extremely tiny. The embryos, zebrafish embryos, are about the size of a penny or smaller than a penny. And you can easily manipulate these and treat them with drugs and have multiple zebrafish in a small area. However, there are some disadvantages to using zebrafish, and that is that some parts of their lymphatic system do not fully recapitulate or mimic or similar to a mammalian lymphatic system. And that's why we primarily use mouse models. So mice are frequently used to generate models of human diseases, and they're useful because you can closely control your experiments with mice, and they're virtually in an unlimited supply. So this is really important, especially when we're working with a rare disease where it's hard to come across patient samples or even patients. We can have a mouse model of the disease and generate hundreds of mutant mice that will develop the disease that we're interested in studying. And in the past, mouse models have been successful in actually improving our understanding of the causes and progression of disease. And they've been useful in identifying new therapeutic targets and for testing existing therapies. However, not all therapies that succeed in mice actually succeed in the clinic. And that's where we have to go back and forth from the bench to the bedside and back again, and use information to further refine and develop our models. So with respect to doing animal work or mouse work, you can't just go in the lab and start working with mice. So I just want to briefly go over the approach that we use on doing preclinical mouse research. So the first thing that we have to do is actually design our experiments. Then we write all of this out and submit what's called a protocol to our institutional animal care use committee. And this is a committee of individuals that makes sure the experiments that you're proposing are ethical and that you've taken in consideration the well-being of the animals. These documents can be extremely long. I have one that's around 100 pages, so they take quite some time to fill out and complete, and they're reviewed. They then, a veterinarian sometimes will ask you to revise your protocol, and then ultimately your protocol is approved. So the experiments that you propose to do are deemed ethical, and it's ensured that the animals' welfare is taken into account, and the mice are going to be treated ethically. We then can either purchase or create the different mouse models that we propose to use. So I think one thing that's not really appreciated is that there are places where you can actually purchase genetically engineered mice. So when we create different mouse models, we deposit these models in different companies, and then other people can then gain access to those mice through those companies. We then breed the mice and then ultimately collect tissues from the mice and analyze the tissues through different methods. And I'll briefly describe those over the next few slides. So in my lab, we've created animal mouse models of Gorham-Stout disease and of generalized lymphatic anomaly. So this is a mouse. What I'm showing here are bones from a normal mouse and our mouse model of Gorham-Stout disease. And this is a bone which has been thinly sliced and then placed on a glass slide and then stained with different reagents. And the stain, in this case, causes the lymphatics to appear brown. So you can see in this bone from a normal mouse, there is no brown staining, and that's because bones do not typically have lymphatics. However, in our mouse model of Gorham-Stout disease, you can see that the bone is filled with lymphatic vessels. We also have been able to show that our Gorham-Stout disease mice lose cortical bone. So what I'm showing here are micro CT images of femurs and ribs, and you can see the normal appearing cortical bone in the normal mice. And in the Gorham-Stout mice, the bones are filled with all of these holes and they're extremely porous. So with this mouse model, what we're doing is we're using it to develop a better understanding of how lymphatic vessels actually invade bone and promote bone loss once they're there. And we think that acquiring this knowledge could help us identify new potential therapies for Gorham-Stout disease. Additionally, we think we could identify new biomarkers to help monitor a disease progression and response to therapy in patients. We've also developed animal models of generalized lymphatic anomaly. And what I'm showing here are lymphatics in the skin. And you can see lymphatics in the normal mouse forming these nice blind-ended vessels. And in our mouse model of generalized lymphatic anomaly, you can see that this very hyperplastic or hyperbranched network of irregular appearing lymphatics. These mice also develop lymphatic vessels in bones similar to patients with GLA, and they also exhibit lymphatic dysfunction. So one way that we assess lymphatic function in mice is you can inject the hindpaws of mice with Evans blue dye, and this blue dye is picked up by lymphatics and then transported from the injection site to lymph nodes. And you can see in a normal mouse, the Evans blue dye is successfully transported from the injection site to the iliac lymph nodes, whereas in the GLA mice, the dye does not make it up to the iliac lymph nodes and stays around the injection site. So we're using those mice to test potential therapies. So those mice were developed in collaboration with Victor Martinez-Glez and Juan Carlos Lopez Gutierrez, who identified somatic activating mutations in PIK3CA in GLA patients. And we developed that mouse model based on those genetic findings. So it's important for patients to participate in research, because by participating in research, that can fuel the development of the next generation of preclinical models. And I think it's important for patients to join registries. The LGDA has a registry And the purpose of this registry is to make the patient community research ready. So the registry can be used to help recruit patients for clinical trials, to learn about the natural history of disease, and to help basic science researchers develop hypotheses that they can then test in their preclinical models. So to summarize a little bit of what I talked about today is that changes to DNA or genetic mutations ultimately cause the proteins that are encoded for by that DNA to misbehave. They don't function properly. These proteins which are not functioning properly causes cells to then not function properly, in this case a lymphatic endothelial cell, and these abnormal lymphatic endothelial cells then form abnormal vessels and ultimately result in disease. So by identifying the genetic mutations that cause CLAs, we're able to generate cell-based models as well as organism-based models such as zebrafish and mice, which we can then use to develop a better understanding of the pathogenesis of the disease and to identify potential treatments and biomarkers for patients. So to conclude this part of the presentation, our understanding of the lymphatic system has increased over 20 years, and however, many questions still exist. But importantly, there are numerous research projects underway to address the basic science and clinical research questions related to complex lymphatic anomalies, and these efforts should lead to a deeper understanding of the causes of these diseases and help identify effective treatments for patients. So I just want to thank you for your attention, and if you have any questions, I just have my email there, and now I'm going to pass this on to Tim.
Thanks very much, Mike. Can everybody hear me okay? Great. Yep. Okay, let me just share my screen here. Okay, can you see the slides okay?
Yep.
Great. Well, good afternoon, everybody. So as Mike introduced you to somatic mutations, I just want to cover that again because it's really important for the work I'm going to show you, and just build a little bit on what Mike's shared with you. So as Mike mentioned, a somatic mutation is an alteration in the DNA sequence of a gene in a cell, and that can then be passed on to the progeny, or as Mike said, the daughter cells of that cell in the course of cell division. But somatic mutations, as Mike shared with you, are different from germline mutations, with germline mutations being inherited genetic mutations in either the sperm or egg that can be passed from parent to child, whereas the somatic mutations, that's not the case. What causes somatic mutations? Well, frequently they're caused by environmental factors, like, for example, exposure to UV radiation or chemicals that can cause mutations, changes in the DNA sequence. And as Mike shared with you, they can affect the function of a protein, the protein is encoded by that gene, then leading to normal function. And somatic mutations are known to give rise to a number of different types of diseases, including cancer. And what we're learning now is that somatic mutations are common in vascular anomalies and especially lymphatic anomalies. So just to show you another kind of diagram of how this works, like Mike mentioned earlier, a germline mutation would be a mutation in the gene, the DNA sequence of a gene that occurs in the sperm or the egg. But these somatic mutations, like for example here, a mutation in the TSC1 or 2 gene, occur in cells after the formation of the zygote or embryo. So mutations in these cells in the embryo can then give rise to mutant cells within the embryo, resulting kind of mosaic tissue where you've got normal cells for the most part, but then somatic mutations in some cells in different parts of the body. So we're learning again that this is the case in lymphatic anomalies, where the somatic mutations give rise to abnormal lymphatic vessels. So in my lab, we use an approach using human cells to study these somatic mutations, particularly the ones that have been identified in lymphatic anomalies. So I'm just showing you here how we put those somatic mutations, those mutant genes, into normal human endothelial cells. So on the left here, you can see that we take the somatic mutation, the mutant gene. It's packaged up in a virus because viruses are very effective at putting DNA into cells. And then we put that virus in the medium that bathes human cells in Petri dishes. So here you can see we've put a mutant gene into these human endothelial cells. The mutant gene has a selection marker, which means that only cells that take up the mutant gene will continue to grow, because we add this drug called purimycin to the cell culture media. So only the cells then that have taken up, here you can see the NRAS mutant gene, will survive. So this allows us to generate large numbers of these cells with this mutant gene. And then the construct that we use inside the virus, the DNA construct, has a switch built into it. We can turn on and off this mutant gene by adding an antibiotic, doxycycline, to the media of the cells. That antibiotic gets into the cells and then switches on the gene that we want to study. And we typically put in a normal copy of the gene as well as the mutant gene having that somatic mutation. So we can compare when we turn on the normal gene compared to when we turn on the mutant gene in these human endothelial cells. So this is a really powerful technique which allows us to basically, like a light switch, turn either mutant gene on or off, or normal gene on or off in these cells. And then we can study how that mutant gene compared to the normal gene affects how the cells signal within the cell and between each cell, how it affects the expression of the proteins made by those cells. Obviously, it'll affect the mutant protein, turn on the mutant protein or normal protein, and then how that affects the cell division. As I mentioned earlier, somatic mutations are often found in tumors and cancer. So cancers, as you know, are caused by excessive unregulated division of the cells, proliferation. So we can study that with our models. And then the other thing I'm going to be sharing with you is how we can take these human cells and put them into mice that are immunocompromised, so we can actually study how the human cells actually grow and how they might cause the actual disease process in humans, but using these immunocompromised mice as a host for the human cells. So this shows you here some pictures of human endothelial cells in these cultures in the Petri dish. And if you go over to the left here, the two panels labeled NODOX, these are human endothelial cells that have the mutant gene, the Q61R, that we've introduced into those cells, but it's not turned on because we haven't got doxycycline in the cell culture media. So this is how these normal endothelial cells look. They look a bit like cobblestones. They're obviously right up against each other. And then below is the kind of the control cells. These are cells we've put the normal gene into. And then when we put the doxycycline into the cell culture media, you can see that 24 hours later, the mutant gene Q61R has really caused these human endothelial cells to change their shape and become very abnormal. And this is what we see in the tissues from patients that have this mutant gene. So basically, we're able to recapitulate what's seen in the tissues of patients in the Petri dish here. Whereas the normal gene doesn't cause that spindling or change in shape of the cells. And you can see that after 2 days, the mutant gene again has caused those cells to look very abnormal compared to the normal gene. So I mentioned that we can also study the proliferation, the division of these cells. So using a really complex piece of equipment called an IncuCyte, on the left here, you can see the proliferation or division of the cells. And you can see that the mutant cells, or the cells that have the doxycycline added and the mutant gene turned on in red here, proliferate a lot faster, a lot more than the cells with the normal gene with doxycycline, or the two controls which don't have doxycycline. On the right, you can see a graph that shows you migration of these cells into a space on the dish. We pull off a small microchamber, and then we can watch and take pictures of the cells migrating into that open space. And you can see again in red here that the cells expressing the mutant gene that we've turned on with doxycycline have a faster rate of migration. And this is often seen with tumor cells or cancer cells, that they migrate a lot faster and invade tissues. So this is some beautiful pictures here that were generated by another investigator here at Cincinnati Children's, Dr. Sandra Schenk. And what she did here was she took glass beads, and the normal cells here are colored in green, and she put the normal cells onto these glass beads so that they grow on the surface of these glass beads, and then she embeds them into a fibrin gel in a Petri dish. And this is scanning confocal microscopy that shows you in green here that the normal cells, obviously they grow around the beads, and then they form sprouts to join up with normal endothelial cells on the other beads. So these cells are expressing that wild type gene, the normal gene, and you can see they form these nice sprouts, and these then become tubes between the beads. But the mutant cells on the right here, which are colored in red, you can see that they don't do that. It's very abnormal. They don't form those nice sprouts. They have a very enlarged structure that's really disorganized. So this again allows us to mimic or kind of copy what happens in the tissues of patients with this mutant gene. And now we have this in vitro model, so it's in culture, so we can study the processes going on here, the disease process, and also test drugs in the system. We can add drugs into the media and see if that blocks these abnormal growth processes of these mutant cells. So this provides a really great preclinical model to test new drugs. So the last model I'm going to share with you, I mentioned earlier, is where we take mutant human endothelial cells, or endothelial cells expressing that mutant gene, and we inject them under the skin of these nude mice. So the nude mice, you can see they don't have hair because they also lack an immune system. So that way we can put the human cells in there and they won't get rejected. So on some mice, we put the cells expressing the mutant gene, and some mice we inject the cells expressing the normal human gene. And then after 8 days, we can take out the plugs. And actually what we do is when we inject the cells, we mix them into gel, Matrigel, and we inject that under the skin. You can kind of see on the left here, there's kind of this bump or lump. That's where the cells have been injected in some gel. You have to put them in the gel so they can grow and form tubes. So this is a pretty complex slide, but I'll talk you through it. So this is 8 days after injecting those Matrigel plugs with the human endothelial cells. And we've removed those plugs. In the top row here, you can see the plugs where the human endothelial cells, where the mutant gene was switched on. We can give doxycycline, that antibiotic, to the mice in their food. So that then turns on the mutant gene. And you can see these plugs are very bloody. They have lots of blood in them, and they're bigger generally compared to the plugs below here, which are mostly clear. They have some blood vessels in them for sure, but clearly not as much as the mutant, the plugs that have the mutant cells injected. And then below are some controls where the mice were not on doxycycline diet. So below here, you can see a graph, and what it's showing you here is the weight of these plugs, which kind of measures how much these cells grew. And then as you can see, there's blood in there, so they formed vessels. So you can see that the cells that had the Q61R mutant gene in them, the human endothelial cells, and the mice were on doxycycline diet, the plugs were much heavier than the endothelial cells that had the normal gene turned on. So we can take those plugs and we can fix them with chemicals and then cut sections through them and actually look at the blood vessels inside those plugs. And that's what you see here. So in this top left corner here, you can see one of these plugs that we've made sections through, and then we've stained it with some dyes to show the structure of these bloody vessels here. And again, this really recapitulates what's seen in the tissue of patients with this mutant gene. And then on the right here, you can see the no-dox controls. On the bottom is the cells with the normal gene expressed because these mice are on doxycycline. So again, this provides a model that we can study both how these mutant cells form these vessels, the types of vessels they make. And then the other thing that we've done recently is start to use this model to test drugs. So this is a kind of in vivo or a mouse model where you can actually take the mice, inject the mutant cells, let them grow a bit. And then in this study here, we used a drug that's already used to treat patients with this disease. We used rapamycin. The drug is known as sirolimus. And so after the cells were injected 2 days later, we started injecting the mice with the drug. And we know that in patients, this will have some effect in terms of reducing the disease progression. The patients show a partial response to this drug. It doesn't cure them, but you can see below that the weight of the plugs in the mice that received the drug, the rapamycin, was lower than the plugs that didn't get that drug, just had the vehicle, which is the solvent that the rapamycin was dissolved in. So in this study, these were all mutant cells were injected. And you can see that the drug that is known to work in patients, at least produce a partial response, works in this model. So this model then we can use to test new drugs, hopefully develop better ones, and also really figure out how the drugs are working in terms of if they do have an effect on the growth of these blood vessels generated by these these cells expressing this mutant gene. So thanks very much for your attention. I hope that was clear. And I want to finish off by thanking the Lymphatic Malformation Institute for funding the work that I showed you. And Mike, as he mentioned, is the director of that, the scientific director of that institute. And as you can tell, we work very closely together. So I think we'd be happy to take any questions now. And my email's there if you want to email me at any time. So Kim, turn it back over to you.
Hey, wow. First off, fabulous. I really appreciate your time. You explained this in such an easy way, and thank you. Does anyone have any questions? If so, please put them in the chat. And until we get started with some questions, I'm going to ask a couple if that's all right. First off, as you spoke, I did hear the word biomarker. Could you both explain a little bit more what a biomarker is and the importance of it?
Mike, you want to go first?
Actually, I was going to let you go because
Okay.
of your angiopotent 2.
Sure. Okay. So great question, Kim. So a biomarker is usually a protein, but it could be some other type of biological product like a lipid or carbohydrate or something that can be easily measured. That's the key thing. Easily measured in patients, like in their blood, for example, and that indicates something about the disease. It could indicate how severe the disease is. It could be used to indicate whether the patients have the disease in the first place. And then also potentially it could be measured while the patient's being treated. So it's easy to measure, as we mentioned, and then gives an idea of whether the patient's responding to that treatment. So we really want to develop biomarkers, and that's what Mike and I spend a lot of our time doing, because we want things that are going to be easy to measure in patients, help diagnose them, help monitor, if they're responding to treatment, and not be invasive, be easy to measure. Mike, do you want to add to that?
You covered it all.
Next, so what biomarkers do we know right now, and how do we look for others? What do we need to do as a community to help all of you?
That's a great question. I'll start and then turn it back over to Mike. So thankfully, funding from the LMI helped us find a biomarker for KLA. And as you know, Kim, this has been fantastic. Angiopotent 2, as Mike mentioned, it's a protein that actually may be playing a role in the disease process. We don't know that. That's part of the work we're doing now. And that can be measured with a simple lab test. And we've set that up at Cincinnati Children's Hospital now, so that physicians from around the country can send patient samples, just some serum from the blood, to the lab here and get it tested for that angiopotent 2. And you can probably share how that helped with your son. So actually, Kim's son really gave us the impetus to get this set up as a test. And it's relatively inexpensive to get it done.
Thank you so much. That's why I brought it up. And Mike D, I want you to talk too, but just quickly, we just sent Tim a specimen of Michael's off yesterday to the clinical lab at Cincinnati Children's. First time doing it that way, easy. We filled out the form, gave it to our, the hospital here, and they've already sent it out. So just to quickly not take over the floor, but by participating and working with these researchers, Michael, without him even knowing it, really did help initiate this as he was getting the treatment of sirolimus plus another drug. And so we were able to follow the angiopotent 2 levels. And just an example of how only one little kid can help. And so you've heard me on Facebook, I'm sure, and maybe sick of it, but it's the idea of all of us as a community coming together, because the more that our researchers have with specimens and blood and participation, the more information we're giving them. Okay, I'm quiet. Mike, did you?
I guess to just sort of say it again, that these biomarkers are extremely helpful and can help avoid the need of a biopsy, which in some cases is needed to really confirm a diagnosis or establish a diagnosis. So this could be a non-invasive way to diagnose a patient and to monitor response to therapy. And, potentially changes in circulating factors could be detected before changes in imaging or other methods could be used. that way we could see really how quickly patients are responding to drugs, and then also if they are not responding, then we could think of different treatments for the patients. But you guys hit it all.
Awesome. We have a fantastic question for you both. How long do these experiments take, and what sort of effort is involved? So I will let you take that away, because as a researcher, I'm with you.
I'll start this time.
Okay.
So for our genetic-based models, we have to breed mice together, and the gestation time for a mouse is about 3 weeks, about 20 days. So after we put the mice together, they breed, and then the offspring from those mice we enroll in studies. And sometimes we can work with very young mice and have results within 3 weeks. Other times it takes about 6 months. So depending on the experiment, I would say our longest experiment was 9 months, and our shortest was about 14 days. No, about 5 weeks. So these can be very long and time consuming, and we're paying for the mice when they're in the animal facility too, to house the mice, to have them food, water, all of that stuff. So I think my average mouse bill is around $5,000 to $6,000 a month just to maintain the mice that we have in the lab. Not even doing an experiment, just to pay for the food, water, and boarding and care.
That was terrific.
Yeah, so that's a great question. I'm really glad to get that question. So thank you, Mary. So since we first discovered angiopotent 2, that was in 2016, and then we published the paper in 2017. So we've been working on these studies now for around 5 years. So it does take a long time to move things through, and a lot of it's dependent on the funding that we can get to do this, how many people we've got working on the studies. So right now my lab is just myself and one technician. If we were able to recruit students, it can go faster, and obviously more technicians, the work can go faster. So Mike and I are constantly applying for grants and trying to get funding from the National Institutes of Health. That's really key, because their grants will give us 4 or 5 years of funding, and that really helps us recruit people and move the studies forward. But just the mouse studies, or the making those mutant human endothelial cells, that took about a year from beginning to end to get that developed. And then once we've got the cells made, then injecting them into the mice, we have to grow up the cells, obviously inject them into the mice. And then those studies were only 8 days long. So injecting the drugs, that's pretty busy work, injecting the drugs. But you could, in theory, get one of those studies done in 6 months to a year, because you've got to generate enough mice to make sure the data's real and significant. So down the road, if we've got enough people working, Mike and I can really move these studies forward, but it does take, years really to make progress. So, but I think we're at a good point now where we have a lot of these models built, right, Mike?
Yeah, so the papers that we've published in the past, it's usually around 3 to 4 years worth of work goes into one publication. That's sort of been our experience. And but like Tim said, we do have models up and running now, so things can move faster now.
Again, you both, as I said, I'm excited to have you, and to have you the opportunity for our community to meet you. This is a comment. I was going to ask it as a question, but you've pretty much answered it, and that is, what can our community do to help advance and accelerate the research? And I think you've really talked about that. It is fundraising to support the research, because we know how long this takes and we don't have the time. And also participating in any way that we can, whether it's giving, going into the registry, the natural history studies and taking part in any way we can. And I think, Dr. LaCroix and Dr. Dellinger did not want to say this, but this is coming from me as a parent, that the research is so critical for us as a community to improve the therapies. And we all have a stake in this. So when we ask of you your time, it's that we all come together, because in the end, it's going to help all of us. Number one. And number two, we would not ask of you to do anything that's dangerous or, take away from your treatment. Absolutely not. And that's really important for you all to take heed in, that we are really aware of how difficult this is from experience. We feel you from experience. And this is our way, an opportunity to pivot and for huge hope in the future. And these two docs are really showing that right now in how much hope there is in what you have accomplished in such little time. So thank you. We have 4 minutes left. Oh, there might be another question. Here we go. This is a good one too. Are there other treatments that seem hopeful? Seems like sirolimus is the main drug. Sure, the spelling is wrong, and I love it. So take it away, guys.
Sure. So trametinib is an emerging treatment for some CLAs. So there have been a few reports or case reports for patients with CCLA that do not respond to sirolimus that have a response to trametinib. So that's probably the next drug on the horizon then for complex lymphatic anomalies. Tim, do you want to add anything?
Yeah, no, I think that's a great point, Mike, that's potentially an emerging drug. And we're going to be testing it in our models too, although it's already being used in patients, as you know, for compassionate use. But the other possibility is combinations. And so Kim, I think, has shared that her son Michael had a combination of drugs. And so combinations may be the future as well, where you can use potentially lower amounts of the drug, so there's less side effects. So that's something, again, we can test in our models to see how those combinations might be working to give better support for using those combinations in other patients.
The other one that there's some excitement about is alpelisib, which is a PI3 kinase inhibitor. And there was a study published a year or two ago, a really dramatic study on where this drug was used to treat cloves, which is an overgrowth syndrome caused by mutations in PIK3CA. And I know that that's another drug that people are interested in.
Okay, there's another one coming up now. But I would like to just go back for all of you. I know we're almost out of time, but I think it's important. These two drugs that Dr. Dellinger and LaCroix have mentioned are, the alpelisib is a chemotherapy drug, so is trametinib, and they're working on targeted mutations. So with alpelisib, it has been shown to, or what it is a against the PIK3CA mutation, and trametinib is what's called a MEK inhibitor. So it's very specific, and maybe the two of you quickly could comment on the importance of that targeted therapy, why we're heading in that direction. And it's because of the research that you all are doing in finding these somatic mutations.
Sure. You could think of these different genetic mutations as being like a gas pedal, and these targeted therapies really being a brake. So it's something that can slow down or shut down these pathways that are hyperactive because of these genetic mutations. So the drugs are really targeted for specific pathways that are active in response to specific mutations. So there's the excitement about alpelisib for PIK3CA mutations and trametinib for mutations that affect the RAS MAP kinase pathway. I think maybe Tim, you want to take a stab at explaining that too?
No, I think you covered most of it, Mike. Yeah, I mean, we're following the kind of playbook of cancer therapy these days, which is using targeted drugs to, once you know the mutation, then you can use a drug that's directed against that mutation rather than drugs that just have very broad effects and can have more toxicity. Kim, I see one more question about biopsies there. That's a really—
Yes, I thought on the heels of this question, this was a perfect one, right? So they asked, are tissue biopsies important in research? And if patients have these tissue, is it important to get it to our researchers? And it's a fabulous question. So you can take it away. I also just quickly want to say that, well, these drugs are chemotherapy, and it's scary to hear that word. I would like to really let you know that right now, what is the hope and positive of that is we're using much lower dosing than what is needed in cancer. So that's a positive. And then the other thing is, by understanding the targets and these drugs, eventually down the road, hopefully there will be this dual, as Dr. LaCroix said, the ability to use more than one type of medication to help our CLA patients.
So certainly the biopsies could be very useful if they're for this disease that we're studying. So like, my lab would be very interested in biopsies from any patients with KLA. And we have to go through a consent process, so you consent to send those to us, but we would be really grateful to receive those. Mike?
Yeah, biopsies are great. They can be used to identify genetic mutations, and they can also be used to collect the cells from the biopsies, and then we can culture those and use those to test various therapies and investigate the molecular mechanisms by which the mutations cause lymphatic changes. So biopsies are always useful.
Okay. Well, we are at time, and just quickly want to thank all of you so very much for participating and being a part of our webinar series and getting us all prepared for the international conference, where we have quite the lineup of speakers, including Dr. LaCroix and Dr. Dallinger. I just would like to remind you that we're on again Monday, August 30th at 12 noon Eastern Standard Time, where Dr. Manuel Carqueo from the Toronto Sick Kids will be presenting Complex Lymphatic Anomalies 101, an overview for us, the layperson. And then to be determined, I still have to get a date, but I will. We are going to talk about treatments and the overview of current and possible future medical treatments, which is great because we just ended with that question for complex lymphatic anomalies. And that's going to be presented by Dr. Kristen Ritchie from Cincinnati Children's Hospital. And we're looking super forward to that. Ooh, finally, remember our conference. Hope you all will attend. The bigger, the better. So we have great questions during the question and answer session. And then just huge acknowledgements to the LGDA. Remember the Lymphatic Malformation Institute, everyone. We are partnered with them, and they are the research branch. That's where all of our research really is being supported right now, and it's important. And then a big thank you to the Rare as One, Chan Zuckerberg, who has made a lot of this possible this year as the LGDA received the grant. So, and with that, thank you so very much. Have a good day, everyone, and hope to see you on Monday.
Thank you, everyone. Have a great day.
Thanks for joining us.